Hello, this is Mohan from Nevizh Private Limited. This video is about our controlled microbial ecosystem, wherein we try to completely replace chemical usage in aquaculture with our biological solutions. So basically, the principles remains the same irrespective of your application, either in hatchery or in nursery or even in growth farms, with minor alteration with application procedures. So we cover two broad problems here: Vibrio management and ammonia management using our biological solution, which can be tracked and can be monitored with easily available toolkits. So these microbial solutions cover wide salinity ranges from 5 ppt to 50 ppt, and especially when it comes to high saline environments, they work phenomenally better than the regularly available bacillus-based solutions, which are basically soil bacteria. So Vibrio management, when we talk about Vibrio management, it's a combination of two products, DriftAct and VibQuench. So each work by different mechanism. And DriftAct is a phage therapy product where we use high number of bacteriophages which target pathogenic Vibrio population. Here in this hypothetical chart, time in x-axis and density in y-axis. If you don't do anything to your aquaculture system, the Vibrio population stays fairly constant. The moment you add bacteriophage population, there's a steep reduction in the total number of Vibrio. This is only for a short time. That's why we clearly name this product as a drift. It gives you a short drift in the population, and you have to act within that window time frame. And that solution is where the phage therapy goes wrong. Is VibQuench. We add a beneficial Vibrio because at aquatic environment, especially the marine towards the saline environment, nothing can compete with Vibrio because the fastest known bacteria to human is the Vibrio itself. So they are very fast in marine environment. So the moment we add DriftAct reduced on the pathogenic Vibrio, we immediately add a brood content of VibQuench, which does two things. It competes for the space and food resources with the other residual Vibrio population here, so that the domination is now achieved by the introduced bacterial population in VibQuench. So that's what we call it as microbial ecosystem control. The product VibQuench works on the basis of QRM quenching, which is basically breaking down the communication of the pathogenic Vibrio. Once you silence the pathogen, they're not going to act upon the shrimp. They're not going to give their toxic. It's going to reduce the toxic gene expression. And it has a evolutionary advantage. Since we are not killing, we are just only cutting down the signal. In this case, for example, acylhomoserine lactone, which is like specific for Vibrio. We are not forcing like in DriftAct. It's going to evolve resistance, but here the resistance chances are fairly less. So we have two bullets. Use DriftAct to suddenly bring down the population of Vibrio, and then use VibQuench to stably have a controlled microbial ecosystem in place. So these two combination works in tandem. While bacteriophage therapy is a better answer for chemical-based microbial control, to get the maximum out of it, few things are to be kept in mind. Number one, always bacteriophage therapy should be followed by a probiotic application, and it should be brewed. And also, frequent application of phage therapy is needed. That's why we have come up with a sustained release technology, especially for hatchery system. We have come up with a sustained release technology for DriftAct. We have also patented the technology. And the number of phage collection is very critical. The product which is having huge number of phage collection has a higher chances of getting success rate in your aquaculture system. Okay, now we pay attention to our ammonia management solution. We call it heteromite, which is a combination of heterotrophic bacteria which consumes not only ammonia, nitrate and nitrate from your aquaculture system and convert that into biomass. So if we have a look, the ammonia is generated by two major sources. One is ammonia being directly excreted via gills by the aquatic animals, and one to degradation of the dead and decaying animals, waste feeds, and everything. These two are the major sources of ammonia in aquaculture. So if you look at the pathway, ammonia is generally converted to nitrite and nitrate through microbial action. These are usually autotrophic bacteria. Their doubling times are very slow. And the ammonia is also in interplay with the ionic form, which is less toxic, but it is pH dependent. And unfortunately in aquaculture, pH is fairly stable. Apart from that, we have the nitrate problem that can be either converted, absorbed by algae, or acted upon by denitrifying bacteria, liberating into gaseous nitrogen. And this whole process is slow. Our answer is heteromite, a combination of heterotrophic bacteria that utilize what we call as an alternative ammonia utilizing pathway. That's how we have patented the bacterium culture also. They utilize all three forms, ammonia, nitrate and nitrate. Just give them sugar, a table sugar as a carbon source. So nitrogen source will come from ammonia, nitrate and nitrate, and then the carbon source from sugar, and they build their biomass. So what we call as a biofloc technology. Okay, we have discussed about our microbial solutions, and then how we apply that to control Vibrio population, and then to do ammonia management. These are not only theoretical answers to the problem that aquaculture people faces. These can be completely tracked and they are traceable because we utilized Vibrio as a probiotic. You can easily track even at the time of brewing or during the application using commonly available microbial culture techniques like use of TC-based plating, etc. For the microbial management system, and also see how the ammonia level rises and goes down with the use of heteromite. You can just see when your ammonia system rises in your tank, and after application you can see for yourself how the ammonia level comes down in your culture system using our heteromite track, which is based on salicylate chemistry to track ammonia concentration in the hatchery, particularly optimized for this particular environment in combination with heteromite. So first time we empower the aquaculturists with the ability to understand what is going on in their aquaculture system. Not only to understand, but to manipulate in a positive way the microbial ecosystem in your aquaculture. Good day.